organization. So this is why I care about a little bit more the interactions that have this sort of larger scale organization. So key behavior of lipids is their strong tendency to form separated phases spontaneously when in contact with water. So in a bilayer, we'll have a polar region, we'll have this hydrophobic region in between. Okay, so for a cellular bilayer, we're typically looking at about 35 angstroms as the distance. This is the length of these two interacting chains. Okay, that's sort of a typical length in the cell. Like I said, you might have a liposome if it's sort of smaller. We're not containing a bunch of cellular structures inside. We could have multi-layer vesicles. Another word for liposome would be vesicle. So we could have just unilamellar vesicles, okay? Or we could have several layers. Okay, this figure here is illustrating a way that you could make these in the lab. So you start with some powdered lipids, okay, and you're going to, it also says they include some sort of lipid-soluble drug. So you're containing something in the nonpolar portion. All right, you put them into an organic solvent, you freeze dry it, and you get this cake here, and then you add your water. So you add an aqueous solution that has a water-soluble drug in it, let's say. Okay? And then you need to sort of agitate it some way or sonicate it, shake it up, and what will spontaneously happen is these multilamellar vesicles. You'll have your drug in the aqueous solution stored in the center, stored in the layers between. You can have your nonpolar lipid-soluble drug contained in the chains. You could control size by extruding them. Okay. And so that would be one method of designing these structures. Okay, we'll talk about hydrophobic effect in like way more detail when we finally get to using the Gibbs equation. All right, but just a couple of questions for you. I showed on the last slide that you needed to do some agitation, do some sonication, and put in some energy to make these things happen. But when we're talking about the biological membrane in the cell, does this structure form spontaneously inside the body? I have some head nods. How do we know? I mean, they're there. We're made of them. It's happening. All of our cells are encapsulated in these cell membranes, and then even inside our cells, and we'll have a figure of this coming up, the organelles are encapsulated in these bilayers. So it happens spontaneously in cells. Where's my mouse? Okay. So we know that our overall change in free energy delta G needs to be negative. Okay? Negative change in delta G is our sign convention for something happening spontaneously. We know, since we're talking about in the body, temperature is going to be relatively constant. There's not a lot of flux there, so 37 C, 310 Kelvin. What is going on with change in entropy here? If we went from individual lipid molecules, or glycerophospholipids, into this bilayer sheet, is entropy going up or down? Down, like way down, right? This is like a really organized structure. Okay, so that kind of seems counterintuitive to the spontaneity. Change in bonding, our enthalpy term here, what are we thinking is happening there? We're making a whole lot of brand new hydrophobic interactions that are giving us some negative contribution. We have the head groups are going to be able to make some hydrogen bonds or polar interactions with each other and also with water surrounding. Okay? But when they were by themselves, the head groups could also interact with the water. Okay? So hydrophobic effect, and we talked about this a little when I talked about protein folding and the hydrophobic core sort of being the primary reason for folding. And that is because if you look at this situation here where you have two nonpolar molecules, there's some number of water that has to sort of, it's kind of held in place around the nonpolar molecule. It's not really able to make interactions that are favorable, but it's sort of trapped there. And then when the two nonpolar molecules are able to interact, there's nonpolar interactions that occur, but what we really get is a whole bunch of free water. And so while this looks really entropically unfavorable, because it looks like entropy is going way down, in reality what we're doing is getting a really big net negative entropy contribution from all this free water. Okay? And we'll talk about this in way more detail in chapters like 8, 9, 10 area. But if you've heard of hydrophobic effect before, I just wanted to kind of draw your attention to the parts of this equation that are contributing to it, and that, not surprisingly, water is sort of the key ingredient here. Okay? So just make sure you understand these different shapes. You are familiar with lipid organization. What do you think are some possible utility of maybe these structures? Why would you want to make a liposome? Why would you want to make a multi-layer vesicle? Ideas? There are no bad ideas in brainstorming. Yes? Drug delivery. Beautiful. I said drug delivery. Now, why might you want a lot of layers?
Drugs.
Different drugs? What else could you do with a lot of layers? Yes? Time release. Slow it down. So you can have some control over time. You have some control over location where it's going to happen. You can put some different drugs in there. Is this biomimicry? Sure is. Okay. So lipids with only a single hydrophobic tail are soluble in water up to a specific concentration. This is called the critical micelle concentration. Okay, so you'll hit a concentration after which they're going to start forming micelles spontaneously. But there's some low level where they will stay. They'll be able to be soluble even in water. Okay, after which you'll get the micelle shape. And so this is supposed to be a cross section cut through the center of it, I guess. The beauty of these drawings is they are so unsophisticated I could make them in PowerPoint if I wanted to. Okay, and then this is a more sort of sophisticated rendering, and again, you can see all the polar groups are this, in your textbook, it's always this red, blue, green stuff, and all the yellow is the hydrophobic tails inside. Okay, these types of lipids are often called detergents, okay, because dirt is hydrophobic, and generally then we can trap dirt inside of our micelles, and that's how you would wash dirt away, such as off of your hands. Okay. So if you go look literally at the back of soap and read some of the ingredients in it, you might see some of these words here, okay? Dodecyl phosphocholine or SDS. These are the types of detergents that are used. And again, it is because when you mix them with water and wash your hands, they're going to trap the dirt inside the micelle through hydrophobic interaction. So one of the things I talked about, I forget at what point I talked about something being like inside out and all the hydrophobic stuff could be on the outside. Okay. So this is a paper called Polar Solvents Trigger the Formation of Reverse Micelles. So what is a reverse micelle? Do you think? Sure. Perfect. It's inside out. Like I just said, all the hydrophobic stuff's on the outside. Now, polar solvents, what do polar solvents mean? Doesn't really make sense that they'd be inside out in a polar solvent. So in fact, that's because they won't be. You need some sort of apolar solvent here to get your reverse micelle shape. So when you have a polar solvent, your typical solvent, you're going to get your normal micelle. You want your hydrophilic stuff on the outside. If you put them in some sort of apolar solvent, you can get a reverse micelle. And in fact, small droplets of a polar solvent, if you drip something polar in, you can kind of control the size, the rate that these form. So just a current relative example of people playing around with stuff as unsophisticated as just lipids in water. Okay. When could something like this be useful in biology or in the body? When might I want to have something that is sort of a reverse micelle shape? Yes. Yeah, if I wanted to be able to cross the bilayer, perfect. So the bilayer has this hydrophobic center to it. Okay, so sometimes to move something through it, I may need to use something hydrophobic to make that happen.